on. Yes. Don't forget to remind me. I won't take offense to it about recording. So we're going to pick right up where we left off. We were talking before, all right, about the circulating hormones, the different types of circulating hormones that are in, kind of mess with this, that's travel in the body, okay? So now we're going to talk about the local hormones, because the local hormones we have, believe it or not, you already have somewhat a brief introduction to the local hormones briefly in 210 when you were studying chapter 2, and we were talking about fats. Okay, chapter 2 is the one that a lot of people, shake their head at. That's when we were learning about atoms and molecules and whatnot. I'm going to switch over here. I'm going to transform. All right. So you're talking about atoms and molecules, and we learned about the different types of macromolecules, fats, proteins, carbohydrates, and then we talked about the phospholipid bilayer, and we kind of segued into this plasma membrane. So here with our local hormones, all right, first, obviously, if they're local, that means they're not going to circulate in the blood. This just won't happen, okay? These guys stay so local, all right? We have this classification of eicosanoids. That's how you pronounce that word, okay? And that is a type of local hormone, and it comes from the fatty acids that are located in the phospholipid bilayer. Remember those, the phospholipids? They had this big circular ball head here, and then these two wavy tails here, and those are the fatty acids right here, those little tails. Okay, all right. So these eicosanoids come from, all right, those fatty acids. Basically, we're going to start off with, all right, I'm going to come right back to this slide, so don't worry about it. This is let me zoom in here. This is where we're going to start off with this guy, big guy right here, Aeroshedonic acid, okay? It's a 20-carbon fatty acid, all right? And it's found in our phospholipid bilayer, all right? And what's going to happen is that fatty acid, all right, is going to be removed from the phospholipid bilayer by this guy right here. Important one to know. Phospholipase A2, all right? Anytime you see ASE on the end of a word, it's an enzyme. Enzymes like to change things. They like to make one substance into another substance. They're important in a lot of our chemical processes in our body. I'll come right back to this slide. Let me go to the previous slide here, okay, for those of you that are writing and typing things down. So what happens here with the eicosanoids, all right, these locally acting hormones, all right, they are going to engage in what we call an enzymatic cascade. So we start off, all right, with substance A, okay, and then an enzyme works on it, okay, we'll say it's enzyme X. And then substance A turns into substance B, okay? And then another enzyme will work on substance B, and that will change substance B into substance C, okay? So that's an enzymatic cascade, all right? So these different enzymes are going to interact with some of these substances in our body, all right? And it's going to create one classification of eicosanoids. They're called prostaglandins. All right, prostaglandins are these locally acting hormones. Leukotrienes are another type. All right, prostacyclins are another type. I'll show you on the next slide some of the different types of locally acting hormones here. All right, but what will happen is that these different types of eicosanoids are going to elicit different types of responses. Some will create inflammation. Some will elicit pain. Okay? So we have these drugs like aspirin and our non-steroidal or NSAIDs, which will block the formation of these eicosanoids. Right? And if we can block the formation of these eicosanoids, we prevent their creation, then we can prevent whatever effect that they're eliciting. Some of these eicosanoids will create inflammation. So you have a drug that blocks its formation, then it's an anti-whatever. So if it's a prostacyclin or a prostaglandin that's pro-inflammatory, if we stop it from being made, then whatever we use to stop it is anti-inflammatory. Okay? So that's what aspirin does. All right, have you ever heard of, give me a COX-1 and COX-2 inhibitors? All right, there's a classification of drugs, and I cannot remember the one that's at the tip of my tongue. All right, it's a very famous COX-1, COX-2 inhibitor, but COX-1 and 2 are enzymes, and so they're involved in this pathway. So if I jump to this slide here, this is kind of giving us, all right, that pathway here. So you got your plasma membrane, all right, phospholipase A2, all right, works on the phospholipids here, and they bust out arachidonic acid. And then arachidonic acid undergoes a series of chemical reactions, that enzymatic cascade, and it yields these guys, all right, prostacyclines, thromboxanes, leukotrienes, a whole bunch of these locally acting, all right, chemicals or hormones. And they can have different effects, inflammation, all right, or pain and whatnot. Asthmatics, leukotrienes are awful for asthmatics because they create smooth muscle constriction. So the smooth muscle that lines the bronchioles, right, the leukotrienes will cause that to occur. And there's drugs out there that we elicit to these patients that blocks the formation of those leukotrienes so they don't have that bronchoconstriction. Okay? So it's really kind of cool. The best, number one, anti-inflammatory drug is, what'd you say? Ibuprofen is good, but I'm talking about something that you need to get prescribed. Well, that's the drug I was thinking of. That's a COX-1 and 2. Steroids. Steroids block the formation right out of the gate. Okay? I don't have a good picture here. That's right. Yeah, Morgan, steroids. They're at the top of the list. So remember on that other slide there I showed you, you got one substance, then another one, and then another one. All right, and they have to go through a series of enzyme changes. Steroid stops it right there. Okay? It's going to affect phospholipase A2, so it stops it right in its tracks, so you never get down to the inflammation area there. Okay? Again, if this was a pharmacology class, I would talk more about it, but this is all I'm going to get into. Okay? So it all starts with this guy here, arachidonic acid, and its enzyme friend, phospholipase A2. Okay? And we go through a series of reactions and we get those locally acting hormones. All right, water-soluble hormones. All right, so if we have something that's lipid-soluble, okay, a lipid-soluble whatever can easily cross the plasma membrane, right? Because your plasma membrane is made up of phospholipids, okay? So if something's lipid-soluble, right, it can pass through the plasma membrane, no problem. But you know what happens when you're cooking and boiling water, if you're making pasta, and you pour a little bit of oil into the water, you can see how that oil doesn't mix in with the water, okay? Well, anything that's water soluble cannot easily penetrate our plasma membrane, okay? So we have to use these membrane receptors, okay? And some of these hormones are water-soluble hormones. So they can't diffuse through the membrane. So we got to figure out another way for these hormones, all right, to elicit their effect on a target cell. Okay, we've got to get it. We've got that hormone's on the outside, and it's trying to get its message to the inside of the cell. All right, especially if it's a cell that synthesizes stuff. All right, beta cells in the pancreas, they make insulin. All right, but we need to get a message to the inside of those beta cells to make the insulin. Okay, so some of these water-soluble hormones need to do that to their target cells. They've got to get their message into the inside cell. They can't get in there on their own. So we're going to now talk about this signal transduction pathway. Okay, so hormone is what we refer to as a first messenger. So it's going to bind on to a cell's receptor. Okay, that's cool. Right? If it was a lipid-soluble hormone, it wouldn't need to do that. It could diffuse right through the plasma membrane, get inside the cell, and do its thing. But we don't have that luxury here. Okay? So the first thing that you really need to focus on is this right here. Activates G protein. I got a cool picture here. I'll show it to you in a moment. All right? We'll walk you through. Okay? But this signal transduction pathway, all right, is what we refer to as the secondary messenger system. And I use this example, and I believe your book uses something very similar to it, okay? I've got a message for one of you, okay? And I'm going to be that person. I'm going to show up at your house to give it to you personally. So I knock on the door, all right? Door opens up. Well, actually, before the door opens up, you have a butler, and the butler says, may I help you? Said, I'm here to give so-and-so the message. So-and-so's in the shower. Can you give me the message? They don't open the door. They don't let me in. So I tell the message through the door to the butler, and then the butler will go and tell whomever I'm going to visit the message. Very similar here, okay? The hormone has its message, whatever its effect is going to be. It cannot enter the cell, so it's got to hand off its message to something else. That's where, all right, the secondary messenger comes into play, okay? But it starts off with this guy right here. We have to activate that G protein, okay? And then the G protein does all the heavy lifting for us because it's going to run around the inside of the cell, and it's going to start randomly activating different enzymes. And as you know, enzymes are going to start to cause reactions to occur. All right, some chemical reactions, but some sort of reaction is going to occur. So this G protein, now that it's activated, it's going to wander around on the inside of the cell, and it's going to activate this. You want to know these two enzymes here, Adenylate Cyclase and Phospholipase C. Okay, these are two enzymes. They're found in the plasma membrane. Okay, and they're just sitting there. They're inactive. They're not doing anything. So an activated G protein will come in and turn these enzymes on. Okay, now once these enzymes are turned on, because what's an enzyme? What is an enzyme? It's a vague question, I know. That's right, girl. Bam! It's a protein. What's a leak channel? It's a protein. Yeah, it's a transport protein. Leak channel is a protein. Sodium ion potassium pump is a protein. As you know, proteins, remember when we were learning about proteins, there's a whole long list of what proteins do. They do a lot of things. All right, this is one of those things. Proteins they'll act as enzymes, okay? Helps with our chemical messengers, our chemical reactions here. So now once we've activated our adenylate cyclase or our phospholipase C, all right, now we're going to create our secondary messenger. Once that occurs, bam, now we can do whatever we need to do on the inside of our cell. We can engage in that chemical reaction, all right, or that process. We can make stuff, or we can turn stuff off, right? If it's like, all right, we've made too much of whatever, we don't need this stuff anymore, we can shut it off, okay? Eventually, we have to stop making insulin. We can't keep producing insulin, all right? Otherwise, what will happen? Go into shock. Your blood sugar levels drop so low because insulin is just plucking your glucose out of your blood, so we got to turn that off, okay? So that's what this stuff can do. The secondary messenger can actually inhibit stuff or it can activate stuff, okay? So this is kind of what I was talking about here. All right, so here are our players, all right, in the secondary messenger system, the signal transduction pathway. One is the water-soluble hormone that can't penetrate the plasma membrane. Okay. 2, we have our receptor protein, and 3, we've got our inactive G protein. Okay. So this is what's going on. Okay. So now our water-soluble hormone, our first messenger, okay, binds onto the receptor. The hormone in this case is the ligand. Remember the lock and key mechanism, right? So you've got your ligand here, it binds onto the receptor, it activates this receptor. So now when this inactivated G protein is just floating around, when it comes in contact, all right, with this activated receptor, because if you notice here, all right, look at the inactivated receptor. See this activation site here? It's a different shape, it's more like a slice, but on this picture here, it's kind of like an indentation. All right, that happens because once the hormone binds onto it, it causes a conformational change, changes the shape of that receptor site, okay, on the inside of that actual protein there. So now that inactivated G protein can attach itself on there. As soon as that happens, all right, this molecule here, guanine diphosphate, all right, GDP, pops off. So it's hanging out on the inactivated G protein. Once that happens, that GDP pops off and GTP, guanine triphosphate, attaches itself. As soon as that happens, bam, we got an activated G protein. Look, it's so happy it runs away. All right, It's activated. It's got all this energy. It's now an activated G protein. It runs away, and it's going to travel down the inside of our cell here. And it's going to go around, and it's going to look for these inactivated enzymes, all right, that phospholipase C, or the adenylate cyclase. So it's going to go around, all right, and this is happening all the time, as long as you have water-soluble hormones that are activating this. So they'll come along. Next slide, please. All right, so it's going to be running around the inside of the cell. It finds the adenylate cyclase, which is not doing anything. And that activated G protein then is going to bind on to the adenylate cyclase here, activating that enzyme. And what will happen is it's going to convert ATP, because ATP is floating around inside the cytosol all the time. All right, mitochondria are making it. Right? You need ATP floating around for all the little organelles inside the cells to do their chemical processes or their jobs. Right? So as long as you're at rest, and it's going to be in abundance. So it's floating around. All right? So it's going to convert ATP into this molecule here called cyclic AMP. And now cyclic AMP is going to float around the inside looking, all right, for different enzymes to activate. In this case, we've got a protein kinase, all right, that's hanging out. As soon as the cyclic AMP binds onto it, we've activated it. What's a kinase enzyme do? What's its job? You guys remember what kinase does? Write this down, make sure you know it. Kinase enzymes like to put phosphate onto things. They add phosphate to stuff. All right, myokinase, you remember from last semester, all right, in chapter 10 about muscles, myokinase, all right, will take a phosphate ion and slap it onto ADP, making ATP. All right, we saw that in muscle physiology there when you're regenerating your energy stores of your muscles. All right, so here you can see that cyclic AMP is now going to activate, all right, for this example, all right, this protein kinase enzyme. Okay? So now what's going to happen with these activated enzymes? Well, now there's a whole plethora of possibilities. All right? So that first one, all right, that first example we talked about was adenylate cyclase. Okay? So that adenylate cyclase, once it's activated, all right, all it's going to do is it's going to create cyclic AMP, and that cyclic AMP will run around and activate, all right, protein kinase A. Right? And protein kinase A is an enzyme that you're going to be using to phosphorylate items so we can engage in certain chemical reactions there. Okay? All right. This slide has a lot of stuff going on in it. So I'll kind of, this is the phospholipase C enzyme. So same story, our activated G protein comes down. It's going to activate phospholipase C, and phospholipase C now is going to convert this PIP2 molecule into either, you don't have to know the actual full names for these molecules, okay? But what you do need to know is these intermediaries, all right, DAG and IP3, and that's all triphosphate, all right? What they'll do is, well, let's start with DAG first. DAG is going to, instead of using the cyclic AMP, DAG will activate protein kinase C, all right? And protein kinase C, all right, is actually going to engage in several different chemical reactions inside the cell, all right? That's as far as you need to know, okay? But the whole point, well, and before I forget, all right, IP3 is going to cause the release of calcium, all right, that's stored in the rough endoplasmic reticulum, right? And then that calcium can go around and also activate certain enzymes. Big picture here is, what I'm trying to explain to you here is these different enzymes, because I don't want you to go and spend all your time trying to memorize this stuff. It's going to be real difficult. Understand, activated G protein is going to activate two enzymes, all right, adenylate cyclase and phospholipase C. And what will happen is with the adenylate cyclase, that will create cyclic AMP, and cyclic AMP will activate your protein kinase A enzyme, in which that will start to undergo, which is used in several different reactions inside the cell. So just the walkaway story from that is it's an activation type of reaction. Okay, we're going to be activating, making things, creating protein and whatnot. All right. In this situation too, all right, with the phospholipase C, instead of using cyclic AMP, we can make either DAG or IP3. Okay, same type of situation. It's going to run around. All right, these activated enzymes are going to go around and activate your protein kinase C. All right, which helps to be utilized in certain chemical reactions, and releasing calcium at the same time too will be helpful for utilizing certain chemical reactions. I don't want you to be concerned about all the different specifics here. Does C happen at the same time? It can. It depends. I see what you're saying. Yes. It doesn't happen all at the same time. What I mean by that is it depends on what the demands of the cell are. Okay. So if it needs calcium, it will release the calcium for certain chemical reactions. As you know, all right, calcium is used in several different types of reactions. All right, for neuron function, for the release of neurotransmitter, also for muscle function. We need it to remove the troponin tropomyosin complex off of actin. Okay. So and also in certain cases, when we're releasing the calcium here, it will open up certain certain ion channels for the influx of certain ions, okay? I just want you to understand that this secondary messenger system is basically utilized to help activate certain processes, and also it helps to inhibit certain processes, okay? So the water-soluble molecule has to activate an enzyme on the inside of the cell to elicit its effect, okay? The cyclic AMP and will help to activate protein kinase A, yeah, which is another enzyme. That's what will act. Well, it will activate the receptor, and then that receptor will change shape a little bit on the inside, allowing the G protein to attach to it. So then the G protein can then become activated. Yep. All right, so here you can see, all right, when we're talking about water-soluble hormone actions here, there's tons of different things that can go on. Like I was saying here, we can either activate something or we can inhibit it, okay? Which is huge, all right, because when we're talking about cell division, all right, we can stimulate growth of the cell and then propagate the actual division, especially when we're embryos, all right? All this, the rapid cellular division that goes on in fetuses, in babies, and in infants, all right? You're going to see rapid cell division, all right? And these water-soluble hormones help with that, all right? Same thing with secretions, like I was saying, insulin, perfect example, all right? This process, the secondary messenger system, all right, is needed, all right, for cellular secretions to occur, all right, when we're making certain protein secretions, all right. Also, when we're talking about the membrane permeability, all right, when we're going to, if we need to add in some things to the plasma membrane, all right, we're going to add in certain cholesterols to actually make the cell membrane a little bit more, what's the word I want to use? I don't want to say rigid, but a little bit more hardy so it can withstand certain stresses, all right? And then we already know this last one here because we learned about that last semester, all right, when we're talking about contraction and relaxation of muscles, all right? So you can see the value, all right, of, all right, these signal transduction pathways, all right? It's huge, especially that last pathway when we're activating the IP3 to release calcium on the inside of the cell. All right, if we can release the calcium on the inside of the cell, and especially if we need to export the calcium outside the cell, all right, for example, don't we need to have calcium in the synaptic cleft and around the synaptic terminals of neurons? Because calcium needs to be in a large abundance, all right, outside the cell compared to inside the cell when a neuron's at rest, because once those voltage-gated calcium channels open up, where does calcium go? Into the cell. That's right. because that's what we want. Because when it goes inside the cell, synaptic vesicles then migrate to the synaptic cleft and release whatever the neurotransmitter is. So this is some of the stuff that we're going to see here. We're just going to start to kind of tie some things together here. All right, so big understanding is basically what I just explained to you is what we refer to as the intracellular, intra being within the cell, enzyme cascade. All right, and it just started, you know, with one substance. An enzyme acted on that substance, converted it into another substance, or activated it, and then it worked on another enzyme. So that's what we're seeing. We're seeing this enzymatic cascade, but one thing that we really need to appreciate is that signal, all right, whatever our signal is, which is either going to be, let's secrete something, all right, let's inhibit something, all right, let's activate something, whatever that signal is, it's going to be amplified at each step through that cascade, okay, which is very important, okay? So the pull away from this slide here is one, the enzymatic cascade is intracellular on the inside, okay, not on the outside, on the inside, and it gets amplified, all right, through each step of the enzymatic cascade there, okay? Oh, and this last part here, cells possess mechanisms to quickly inactivate, all right, the intermediate. That's important. We got to shut this machinery down, all right? Remember, when we were talking about the nervous system, all right, the nervous system, all right, it can elicit quick responses, right? I can elicit a quick response in this room by shutting the light switch off. I make it darker here. That's immediate, all right? But with the endocrine system, once we've made those hormones and they're floating around in our circulation, they just don't magically leave the circulation, all right? They're going to elicit their responses for a prolonged period of time compared to the nervous system, all right? So when that happens, all right, when we need to shut this machinery down and end that, all right, what we can do is that whole cascade that we just I showed you in those 3 slides there, all right, if we can stop the production of any of those what we call intermediates, any of those parts, all right, or pieces, if we can stop the production of cyclic AMP, or if we can stop the production, all right, of IP3, all right, if we can stop any of that, all right, then we can stop, all right, that secondary messenger system, and then we won't be producing, if, for example, if we're trying to produce insulin, then we can shut that down. We'll stop producing insulin, all right? So a good way to do it is, let's break down those secondary messengers. A good way to stop a muscle contraction, there's several parts. If you recall from 210, all right, if we break acetylcholine down, that's the neurotransmitter, we use an enzyme for that, acetylcholine esterase, okay? If we wanted to prolong a muscle contraction, we could prevent the breakdown of acetylcholine by giving somebody a drug that destroys acetylcholine esterase, the enzyme that breaks down the neurotransmitter. The neurotransmitter will stay in the synaptic cleft longer, sustaining our muscle contraction. See what I'm saying? So we can stop certain things or prolong certain things. In this case, if we want to, a good way to control our signal pathway to shut it down is, let's stop making cyclic AMP. Boom, stopped it, okay? We won't make any more insulin. So the water-soluble hormones are only inside the cells. Well, the water-soluble hormones are Brittany, great question. The water-soluble hormones never get inside the cell, okay? Because the water-soluble, they can't diffuse across the lipid. Remember, fat and oil does not mix, right? So they can't, the water-soluble can't cross over the plasma membrane. So they've got to utilize something on the inside of the cell. That's the butler there, okay? So that butler, that G protein or any of those activated enzymes on the inside, like the cyclic AMP, okay, what will happen is they're going to carry that message onto the inside. You got it now? Sweet. I love it. Love it, love it. All right, so this is basically, all right, another concept that I want to talk to you folks about hormone interactions, okay? And we see this quite frequently, and I'm going to give you some of the basics, and then hold on to your hats, because then we're going to get into the basic, excuse me, not basic, but then we're going to get into the individual hormone. We'll talk about growth hormone. We'll talk about thyroid hormone. We'll get into all that fun stuff, okay? So what you want to understand here with our hormone interactions, all right, this slide is showing you've got these different types of target cells, but on these different types of target cells, you've got different receptors, okay? But some target cells will have multiple receptors. So you can see all the different receptors on this adipose, liver cell, and muscle cell. And then this example target cell has a bunch of all the different types of receptors, okay? So if you have different hormones floating through your circulation, because you can have target cells that be affected by many different hormones, all right? So there's a couple different types of interactions we can have. The first one is what we call synergistic, all right? This is when these hormones that affect the target cell work together, and the response that they create is going to be larger. The sum response will be larger than what the response would have been with each individual hormone acting on that cell. I'll give you an example here in a second. The second one is permissive. So you're going to need one hormone, all right, has to be present, all right, first, and then the action of that is going to be followed by a second hormone. Perfect example, let me hold that thought. I'm going to jump into the examples here in a second. And then finally, antagonistic. I think we're pretty familiar with antagonistic, all right? The effects of hormone A is opposite the effects of hormone B. Insulin and glucagon are perfect examples. Insulin decreases, all right, blood glucose levels. Glucagon increases it. Okay, they'll be opposite. Okay. So this next slide here. Oh wait a minute. I don't know why I threw that in there. Did I skip a slide? All right. I must be losing my mind. Hold on. There we go. I did skip a slide. Shame on me. Naughty boy. Yes, ma'am.
That synergistic says that one hormone is actually given but isn't that what?
No, you need, and I'll explain the example of the permissive one. All right. So this one's perfect because we talk about estrogen and progesterone. Together, their effect on the target cell will be greater. The sum of both of their effects will be greater than if just estrogen or progesterone worked on that target cell. All right, and the perfect example would be in hyperplasia of the uterine wall, all right, of your uterus there. Okay, if only one was acting on it, all right, the effects would not be, the response would not be as effective. Now, go ahead. Exactly, but also, that's a great question, also birth control drugs have to do with at certain points of the menstrual cycle, all right, of the typical 28-day, you'll have increases of one hormone versus increases of the other. So it helps with that also. But yeah, that's a great question. All right, so with permissive actions, all right, this is a great example. Breastfeeding, okay? You use oxytocin and prolactin, okay? But for breastfeeding to occur, prolactin has to actually first, one, create milk production. So if we don't have prolactin, oxytocin is useless because oxytocin for breastfeeding ejects or expels the milk. So if we don't have prolactin making the milk, then oxytocin is useless because it won't eject the milk because we won't have any made. So we need prolactin first to make the milk, and then oxytocin will eject the milk. And then antagonistic interactions, I talked about that with glucagon and insulin, all right? They're just opposite of one another, okay? All right, so this should look familiar. I'm going to kind of quickly go through just a quick review here. We talked about this in lab, all right, on Monday, all right? We're going to now go over, all right, some of the glands here and tissues that are going to be important, all right, when we're talking about the endocrine system. One, you've got the master gland, the hypothalamus. We all know where that's located. What part of the brain is the hypothalamus found in? Remember, there's 4 parts to the brain. Brainstem, cerebellum, booyah, diencephalon. That's right. There's 3 parts of the diencephalon. Hypothalamus is one. Epithalamus is another. That's where our pineal gland is. All right, and then the thalamus. Okay. So the hypothalamus is going to exert its control over the pituitary gland. All right, this is kind of like what I was saying on Monday. All right, our hypothalamus is the captain of the ship, and our first mate is the pituitary gland. Okay, and then the pituitary gland is going to affect all these other structures down here. We're going to go through that. Okay, all right. So we talked about the pituitary gland on Monday. Another name for it is the hypophysis. All right, I'm joking. Hypophysis. I keep thinking hypothesis. Hypophysis. We know where it's located in the sphenoid bone and the sella turcica. Okay, for the pituitary gland, all right, it is going to do a lot of the heavy lifting for the endocrine system. Not to say that the hypothalamus doesn't, all right, but the hypothalamus is connected to the pituitary by the infundibulum here. Okay, so we have a front and a back, the anterior and the posterior. Okay, the posterior is the neurohypophysis. Okay, and that's when we were talking about those two hormones that are produced in the hypothalamus, and then they wind up down in the pituitary gland. We'll get into that. And then the anterior pituitary is known as the adeno or adenohypophysis. All right. So here's the posterior pituitary, the neurohypophysis. All right. So we're going to see these neurons, all right, that are going to project from the hypothalamus down into the posterior pituitary. The cell bodies, or the somas, remember that term, soma, all right, are going to be, you need to know this, all right, are going to be located in this first nucleus here, the paraventricular, and then our supraoptic. Remember SA, all right, OP, not soap, but close. All right, the supraoptic nucleus is going to produce ADH, which is the antidiuretic hormone, also known as vasopressin. We'll talk about that in a second. And then the paraventricular nucleus is going to produce, all right, what hormone? Begins with an O. I just talked about it. Oxytocin. Yeah. Okay. So remember this. I can almost guarantee you it'll be asked on a test. All right. 7 out of 10 of you will have this on your test. All right. Oxytocin, all right, and ADH are both made, synthesized in the hypothalamus. All right. Produced is another synonym that you can use. All right. But they are released, secreted, all right, from the posterior pituitary. Okay, don't get that confused. So if you're taking a test and it mentions oxytocin or I was gonna say ADH, yeah, what did I say? I was gonna say ADHD. I was thinking of attention deficit hyperactive disorder. All right, but antidiuretic hormone. All right, ADH. If you see that in a question, you better darn well look to see what the actual verb is that they're using. If they say synthesize, produced, then your answer should be hypothalamus. If it says released, secreted, all right, then you're going to think posterior pituitary. I see it happen all the time. All right, so just be really, really on top of that. Sorry? Yep, essentially it's made. Any synonym that you can, match to that, it's made in the hypothalamus and it's secreted or released from the posterior pituitary. Okay, so the axons then, they leave from the cell body and they're going to go down, all right, to the synaptic knobs. All right, the axons are in the infundibulum, all right, but remember, tract, bundle of axons, all right, we refer to that as the hypothalamo-hypophyseal tract. Okay, and then at the far end, the synaptic knobs, the pars nervosa, and that's going to be in the posterior pituitary. All right, and that's where those hormones will be released from. All right, so that you've seen this picture already. Okay, this is just showing you the two different nuclei here, and then how our axons are traveling down into the posterior pituitary area here. Okay, antidiuretic, ADH, or oxytocin, all right, are both going to be released from that area there. All right, again, this slide should be familiar too from Monday. All right, anterior pituitary, and in this situation, think of the posterior pituitary dealing with nerves. That's why we call it the neurohypophysis. All right. The anterior pituitary, which is our adenohypophysis, is going to deal with blood. Okay. Circulation. Okay. So its system is referred to as the hypothalamic hypophyseal portal system. Okay? So that means we're dealing with blood vessels here. Okay, remember we had the primary plexus, the secondary plexus, and then we had our portal system, our portal veins. We'll talk more about that when we're going to learn about the different types of veins and venous systems when we talk about circulation. We'll talk about a portal system, right? But the primary plexus is located up in the hypothalamus. The secondary plexus is going to be down in the adenohypophysis, the anterior pituitary. And then in between, we have our portal vein system there, okay? So essentially, they're just going to drain the blood from the hypophysis into the anterior pituitary. Okay, so that's how, all right, the hypothalamus communicates with the pituitary gland. One of two ways, all right? It's going to communicate through the nervous portion of it, all right, or through the blood stream here. So the hypothalamus will release, all right, the classification here, all right, think of the hypothalamus, all right, and I talked about it. It releases what we call regulating or regulatory hormones. And basically, these hormones are the bossy hormones. They tell, all right, another tissue what to do. And that's what it does to the pituitary. I feel awful for the pituitary gland because it's going to tell the pituitary, hey, pituitary, all right, you're going to release this, or you're not going to release this, okay? So that's what it does. So it's going to regulate what the pituitary can and cannot do, all right? And we'll get more into that. So you can see, I already showed you this on Monday, all right, this list of different, all right, hormones that the hypothalamus releases and what the anterior pituitary releases. And notice that some of these are what we call releasing hormones, and some of these are, where are we, inhibiting hormones, okay? Yes. All right, but what I mean, but when we go through these different hormone axes, all right, or you'll understand why. You'll understand. Yeah. So, pardon? Yeah, What was that figure I told you? Was it 17.21 or 1712? I have it. Hold on, I'll find it for you. This guy, right? Yeah. Oh, it's in here. This guy, 1712. That's the one. Okay. This is the one that if you can convert into flashcards, it really will help you. We're going to go through this in more detail. I've had students do this, and they do really well in understanding this by converting this figure into flashcards.
The other chart at the beginning of the chapter.
I'm trying to remember which chart that was. What did it look like?
Yeah, it's like 2 pages.
Oh, yes, yes.
My previous professor wanted us to memorize those, and I failed the class.
Well, yeah, okay, that one that you're talking about.
Yeah.
Yes. And I've had students, and again, you can make that into flashcards too. I like this one, it's much more simpler.
Okay.
But here's the thing, we're going to go, that condenses a lot of what I'm going to talk about over the next 2 lectures, all right, including this one. So you'll kind of understand that. So when you're done with chapter 17, when you go back and look at that chart, and if I've done my job pretty well and you've done your job pretty well, you should understand that chart no problem. Yep, you should breeze through that, okay? So, and I'm going to, like I said, I'm going to take it nice and slow, all right? We're going to go at a nice pace here. I'm only blowing through this stuff quickly here because we've already went through it in lab. So this should be reviewed. Let me go back to, all right, I jumped too far. Okay, so let's now talk about, okay, some of the interactions that we're going to see, all right, between our master gland, all right, and we'll start off here, all right, with the posterior pituitary, okay? So again, this first, all right, enlarged print here is basically telling you that the posterior pituitary is a storage and release site for the hormones that the hypothalamus made, okay? So you have these specialized cells in the hypothalamus, these neurosecretory cells, that made this hormone, and then it's got to ship it off down to the posterior pituitary. Now, you do want to know this. It gets down there through this process called fast axonal transport. Okay, there's fast and slow. All right, but that's a basic true-false question kind of scenario. Just know that it gets there fast. All right, slow axonal transport takes a little while longer. All right, and when it comes to eliciting certain effects, all right, for hormones, all right, we know that it takes a little bit longer for the endocrine system to have its effect occur because it's got to release hormones, circulate hormones to the bloodstream. That takes some time. Whereas when we're dealing with the nervous system, stuff happens fast, all right? So these two hormones here are going to be stored in the synaptic knobs, okay? And so when those neurons fire, they're going to release, all right, that antidiuretic hormone, also known as vasopressin or oxytocin, much similar to a neurotransmitter. How that was released, you know, when we saw back in chapters 10 and 12, all right, that neurotransmitter, when it released, similar way, all right, here with our hormones, okay? So that antidiuretic hormone, now look at the term, antidiuretic. Diuresis is going to be urinating stuff out where you're pretty well hydrated. So this hormone is going to prevent OT and then you release from the synaptic knob. Yeah, yep, they're going to be trapped there. Yes, all right, they're released from the synaptic knob, okay, into the bloodstream nearby, and then they'll enter the circulation. Okay. So basically, this hormone prevents you from becoming dehydrated, right? So what happens when you're dehydrated? When you're not going to be peeing a lot, right? So this is going to decrease the amount of urine that you make. But guess what? Doesn't it make sense that it wants to stimulate thirst? Why? Exactly. Makes me thirsty. I'm going to drink some water. Now I'm rehydrating myself. Okay? So I want to walk you through some of the stuff so you're not trying to memorize everything. If it makes sense to you, then it's going to be easy for you to recall that information there. All right? And then it also, this is important, it constricts the blood vessels. Okay? That's one of the processes. That's why it got this name here. Vasopressin. Vaso being blood vessel. Okay? Pressing in. All right? That's constriction. Okay? Don't think about it too much. All right? Oxytocin, we're going to talk a lot more about oxytocin later on when we get into the reproductive part, near the end of the semester here, all right? But you need to understand that uterine contraction, big time, okay? So when baby's trying to come out, when the baby's head is pushing on the cervix, all right, there's receptors there that are going to stimulate, all right, the release of oxytocin to cause more uterine contractions, all right? Yes, ma'am? You go, girl. That was my next point. You had a good teacher last semester. I don't know who it was, but I want to know. Yes, that is the positive feedback loop. Yes. All right. So that's what you're going to see here. All right. We already talked about milk ejection. Okay. When we're talking about prolactin, and then also we'll talk more about this about the emotional bonding too. All right, during the nursing part. People don't talk about it as much, because uterine contraction and milk ejection get all of oxytocin's attention. But emotional bonding is key, okay? So now let's jump into the anterior pituitary, all right? That's going to be our blood system, all right? So like I was saying before, the hypothalamus, all right, is going to release what we call regulatory hormones. And there's two classifications. There's going to be releasing and inhibiting. But just understand that its basic job is going to be to tell others what to do, okay? And so when it tells the anterior pituitary what to do, all right, that anterior pituitary hopefully will release some of its hormones into, all right, the general circulation. And if it doesn't, all right, it's because it was told not to by an inhibiting hormone, okay? All right, so real simple. Okay, it's what we call a binary relationship. All right, it's like a light switch. It either turns the light on or it turns the light off. That's what your hypothalamus will do with the endocrine system here. It's either going to tell the anterior pituitary to release something, or it's going to say don't release anything. It's inhibited. All right, keep it simple. Keep it simple. KISS principle. Okay. All right. So here are the two types. You got your releasing hormones and the inhibiting hormones. Don't worry, we're going to go through all of these, all right? You can write them down if you want, all right? You don't have to, all right? There's a lot more of the releasing hormones than there are of the inhibiting hormones, all right? But it's real simple, all right? The releasing hormones are going to increase, all right, the secretion of the anterior pituitary to release its hormones, all right? And if it's an inhibiting hormone, released by the hypothalamus, it's going to decrease secretion. Real simple. Let's keep it easy. All right, because we're about to get into some stuff here. What's our time looking like? All right, not too bad. All right, we're about to get into some stuff here that is going to be a little bit, I'm not going to say more complicated, it's just going to be a little bit more detailed. All right, but I promise you, I will walk you through it. All right. All right, everybody at home, how are you doing? Am I going too fast for you? Sometimes when I get excited, I go fast because I do enjoy talking about this stuff. All right, I hope you're all awake and paying close attention to me. All right, you're good? Morgan's good. All right, then we good. All right, so we're going to go into a little bit more detail with some of these hormones here. All right, Caitlin's good too. All right, so if you look at the names, first of all, to really kind of decrease confusion on a lot of things, really just take a moment as you're reading the stuff and studying it to look at the hormone name. The first one up there, thyroid stimulating hormone. What do you think that hormone does? Oh yeah. I mean, let's not make it more complicated than what it is. It's going to stimulate the thyroid, all right, gland to release, all right, thyroid hormone, okay? And we'll get into some of the details about that, all right? So where is it going to release it from? The thyroid gland, all right, located inferior to your thyroid cartilage, all right? So prolactin here, okay, this hormone here, all right, causes milk production. We talked about that earlier, all right? And it's also going to cause, all right, once mom has baby, all right, she'll start, even before so, she'll be producing prolactin, all right? And that is going to start to enlarge a lot of the glands in the breast, you know, and as mom is starting to produce milk there, okay? So we'll see an increase in the size of the glands there, all right? So we can have two hormones that are going to affect prolactin, okay? One is obviously, all right, prolactin releasing hormone. I think we can all figure out what that's going to do, but I'll say it. I'll be the person. It's going to cause prolactin to be released from the anterior pituitary. Or we can have prolactin inhibiting hormone, and that's going to decrease the secretion of prolactin. All right, we'll talk more about that. This next one here, the adreno, if we stop here at adreno, all right, what gland is that going to affect? The adrenal glands, okay? So again, some of these titles will tell you what tissues that we're dealing with, what glands that we're dealing with, okay? So the adrenocorticotropic hormone, also known as ACTH or corticotropin, I'm going to call it ACTH because if I have to keep saying adrenocorticotropic hormone, my mouth's going to dry out, okay? But what this hormone is going to do is it's going to cause the release of cortical steroids, which is a group of hormones, there's about 25 of them, that are going to get released from your adrenal glands, specifically the outer portion of the adrenal glands, which is called the adrenal cortex. Okay, so that's where this other part comes from. Cortico, all right, referring to the cortex. Okay, so adrenal cortico, all right. So its release is triggered by, all right, corticotropin releasing hormone. Okay, and obviously that releasing hormone comes. So we're going to get into more detail. I'll show you pictures. What I really like about our textbook is that it does have some really good illustrations. I'm a picture person, all right? Even though I can't draw with a darn, so you know that, all right? But I will do my best, all right? Just a couple more here, all right? So the next classification of hormones are the gonadotropins. If you look at, again, the beginning portion of this, gonad, all right, that's going to be our sex organs. Okay, testicles in men, all right, uterus and ovaries in ladies, okay? So there's two types of gonadotropins, all right, follicle stimulating hormone and luteinizing hormone, okay? So that is going to be triggered by the gonadotropin releasing hormone from the hypothalamus, and then that will trigger the release of one or both of these hormones, FSH or LH, okay? So what happens in the female? All right, Ovarian development, we'll see a lot of this in later chapters. We'll get into much more detail, and we'll see the effects of estrogen and progesterone and all that. All right, and also it's going to affect the release of estrogen and progesterone during the menstrual cycle. All right, we'll see a lot of that going on too. Okay. In men, easy. Makes sperm and makes testosterone. Okay. Men are basic. We're simple creatures. Just kidding. Sometimes people think we're too simple. All right. Should we make flashcards for these? Kayla, yes, I would make flashcards. Again, start off with figure 1712. All right, and if you want to make flashcards for those big long tables in the beginning of the chapter, feel free. I had some students do that too. But there's a lot. There's a lot in there. All right, growth hormone, one of my favorites, also known as somatotropin. All right, growth hormone is going to trigger the liver to release these two substances here. All right, insulin-like growth factors 1 and 2. And insulin-like growth factors 1 and 2 will work synergistically. We now all know what that means. It will work synergistically with growth hormone, okay, and help elicit all the effects of growth hormone, because there's a lot of stuff that growth hormone likes to do, okay? So we'll see, all right, not only will it stimulate cell growth, it also stimulates cell division, all right? As we're growing from a little kid to an adult, all right, we're going to see a lot of cell division, all right, if we want to make our bones grow, all right, our muscle tissue grow, okay? All right, we already saw that. I'll skip that. All right, on to our very, very first hormone specifics. All right, growth hormone, GH. Okay, we're going to talk about, all right, how we regulate its release. Okay, and we're going to talk about what it does. Okay, so the first thing is we're going to start in the hypothalamus. Okay, so what is going to regulate the release of growth hormone? Well, the hypothalamus is going to release either growth hormone releasing hormone, and then that will therefore stimulate the secretion and release of growth hormone, or the hypothalamus is going to release growth hormone inhibiting hormone, right? And that will decrease the release of growth hormone, all right, into circulation, okay? So that's a binary thing, all right? So keep that in mind. So the hypothalamus is either going to encourage, all right, the release of growth hormone, or it's going to discourage it, all right? So you do want to know these, all right, everything I say you want to know, but all right, you do want to know, all right, what is going to impact, all right, growth hormone release, okay? Specifically, what's going to what's the stimulus? Remember the reflex arc I talked about? I just taught it to my Bio 210 class last night, all right? Remember the homeostatic mechanisms? You have a stimulus, you have a receptor, you have a control center, and an effector, all right? That is going to be, all right, our template for learning all of these hormones there. So we're going to have a stimulus. So here are our stimulus, or excuse me, stimuli, okay? How old you are. The younger you are, the more growth hormone you're going to release. All right, I'm over the age of 30, so I'm not going to be releasing too much compared to a 10-year-old. Okay, so I'm going to show you a chart here, but as you get older, your release of growth hormone goes down. Okay, the time of day, believe it or not, the time of day that you release growth hormone is important. All right, so that's one of the reasons why. Well, I won't get into that. I'll talk about that later. Nutrient levels. I can't grow things. Do you think that people can build a house if they don't have materials to build a house with? How can I expect someone to build me a wood house if I don't give them any wood, right? Would you agree with me? All right. So if you don't have enough nutrients in your body, all right, what do cells like to use for nutrients? Spit something out at me. Carbohydrates, glucose. What else? Proteins, fatty acids, all right? Those are nutrients. If you don't have nutrients, all right, then you're not going to be able to build stuff, okay? So we definitely need adequate nutrient levels, all right? stress and exercise. And I say stress and exercise because they're going to stimulate that release of growth hormone. If you remember this, the law of supply and demand, if there's not a demand for something, there's really not going to be a supply for that. So there's a demand, like if I'm exercising in the gym, working out, all right, my muscles are going to demand, all right, more protein to make more contractile units, more sarcomeres, because it's going to want to make more actin and myosin cross-bridging, all right, myofilaments there, okay? So if the demand is there, then the supply will be there. I lost connection, sorry, okay, we were waiting on you, Brittany. We were waiting here like for 5 minutes for you to get back. All right, just kidding. And the nice thing is you won't have to worry about it. You can always go back and watch it in the video. All right, so let's talk about some of the effects of growth hormone. I'm going to tell you something. I'm going to spell this stuff out to you, and I'm praying that it'll be easily understood. I'm going to try to make it as easy to understand and as, how do I want to say it, as matter of fact as possible. Okay? So here's what's going to happen. Growth hormone gets released, it goes into the circulation, and some of that, if not all of that blood, is eventually going to pass through the liver. Okay? So it's going to go to the liver, and it's going to stimulate the release of, we talked about those guys earlier, all right, the insulin-like growth factors 1 and 2. Okay? Now, insulin-like growth factors are very similar to our growth hormone. The nice thing is they last longer. So that's cool. All right? And they have that synergistic relationship. So we'll get more bang for our buck. All right? So this fact I love, that all the cells in your body, all of them, every single one, has a receptor for growth hormone, all right, and IGF. Cool, I think, all right? Because what we're going to see is this growth hormone and these IGFs are going to increase protein synthesis. That's huge. We use proteins for everything, right? I mean, if you want to, you can go back to your notes from 210 and see everything that proteins are utilized for, all right? Half of the weight composition of a plasma membrane is made up of protein, not to mention all the protein stuff that's floating around inside the cell, all right? The majority of your muscles are made up of, all right, protein, okay? So we use that to increase the protein synthesis, all right? Also, think about it, growth hormones in high production in our younger years when we're doing a lot of growing, so we're going to use it for cell division, and then of course cell differentiation, when cells are transforming or turning into or developing into another type of cell or a more mature form of that cell type, okay? Like red blood cells, for example. They start off as known as reticulocytes, that's the baby red blood cell, then they eventually mature up to what we call an erythrocyte, okay? We'll talk a little bit about that. All right, so this next part here, when you look at this next part here, you're like, oh man, what are these funny words? I don't remember these. All right, but I'm going to break them down for you, okay, because that's what I do, okay? But the important thing, all right, with growth hormone, all right, is this concept here, all right? It's going to release nutrients from storage. So star that, put that at the forefront of your mind. So any nutrient that is in storage, and we just named off a bunch of nutrients, proteins, all right, carbohydrates, glucose, all right, fats or fatty acids, all right, all of that stuff, okay, all those nutrients, we're going to release them from storage. We want to get them into circulation because they're not doing us any good when they're being stored away, all right? Like I said, somebody that's building a house needs a supply of whatever. All that stuff, all right, if they want to build that wood house, that wood is not doing them any good at Lowe's, okay? They need it there on site, okay? So that's what we're going to do. So this first term here, glycogenolysis, I'm saying it too slow, glycogenolysis, okay? What is glycogen? Bam. I can't really see who said that because they're all wearing masks, so I just heard it from over there, all right? But yeah, it's a stored form of glucose. Glucose is what we consider a monomer, and then glycogen is when we string all these monomers together, okay? here we go. Going back to 210. What two organs or tissues have glycogen? Liver. I can't tell who's talking. Liver and skeletal muscle. Skeletal muscle. Need to know that. Need to know that. So glycogen is going to be found in the liver and skeletal muscle. Okay? So glycogen is the stored form of that. Lysis means to break down. So basically, this is the breaking down of glycogen. So if we're breaking glycogen down, that's stored in the liver and in the skeletal muscle. Most of the glycogen in the skeletal muscle will be used by that muscle. Okay? But the liver, when it's breaking down its glycogen, what's it going to do with that glycogen? Or what's it going to do with that glucose now? Right. So it's going to dump it into the blood. It's going to start just dumping it, offloading it into the blood so it can circulate throughout the body. What's the number one organ in your body? Who gets first dibs on glucose? That's right. The brain. Keep that in mind. Brain gets first pick. The brain prefers glucose. You know, if we run out of glucose, there's other things it can use, but it's important that you have glucose in your body. Okay? So brain always gets first dibs. Okay? So glycogenolysis, the breaking down of glycogen, is going to be stimulated. We want to what? Release nutrients from storage. The next one is gluconeogenesis. Gluco is glucose, neo means new, right? Genesis means to create. So we're making a new form of glucose, we're making new glucose from non-carbohydrate sources. Fatty acids, that's a non-carbohydrate source. So is proteins, all right? There's a couple others, but let's just focus for right now. So, again, we're going to make more glucose. We're going to release more of these nutrients from storage, so we're going to stimulate gluconeogenesis. Okay? So that might mean that remember our adipose tissue? The predominant cell type in adipose tissue, what's it called? Do you remember? That's right, an adipocyte. An adipocyte. Okay? So the adipocyte, its job is to store fats. Okay? So what that adipocyte is going to do is it's going to break down the stored fats and release that stored fat into your bloodstream. Because again, we need nutrients circulating. So the process of breaking down the stored fats in an adipocyte is called lipolysis, right here. Lipolysis. We break down those stored fats, all right, triglycerides. Triglycerides are made up of a glycogen backbone with 3 fatty acids attached to it. Real quick, fatty acids come in different sizes. We have short chain, long chain, medium chain. Okay, the longer the fatty acid, the more energy. Fats are the best form of energy that our body has available. So the longer the fatty acid, all right, the more energy we can get from it. So we're going to start to break that stuff down. All right, so here are the 3. That's a messy. 1, 2, and 3. Those are all the processes that we stimulate because they're going to dump the nutrients into blood. So the cells that need to do their thing, that need to differentiate, that need to divide or whatever, need to make something and build stuff, they have that energy. So now, at the same time while that's occurring, we are going to stop making glycogen, okay? Because to make glycogen, we would have to pull glucose out of the bloodstream. We don't want to work against ourselves, okay? So we're going to inhibit the making of glycogen, and at the same time, we're going to inhibit the making of fats, or our triglycerides, okay? So keep that in mind. We're going to inhibit, because remember, we're busy putting nutrients into our blood. You do not want to take the nutrients out of the blood. So we're going to stimulate the processes that put stuff into the blood, and we're going to stop those processes that take our nutrients out of our blood. You guys with me on that? Are you with me? All right. Come on. It's important stuff there. All right. Yeah, you are with me. Yes. I feel like, what's his name, Mel Gibson in Braveheart. Freedom! But in this time, we'll say endocrine, growth hormone. All right, so this picture here, let me go over this with you. All right, so we're going to go, again, think of that reflex system, that homeostatic system I was telling you about. Stimulus, receptor, control center, effector. All right, so it starts off here with the stimulus, and I already went through all that. All right, how old you are, the time of day. Also, I have to zoom in here. I need new glasses, I can't see as well. All right, also, the important one that's listed at the top here, negative feedback by growth hormone. All right, remember there's two feedback loops, positive feedback, which we already talked about, all right, and the negative feedback, positive feedback moves in the same direction of the stimulus, okay? Negative feedback, the effect is always in the opposite direction of the stimulus. Perfect example, body temperature. When it gets too hot, all right, your body works on trying to make things cooler, okay? So in this situation, when there's a decreased amount of growth hormone circulating, all right, the negative feedback is going to kick in, and we're going to start to increase the amount of growth hormone circulating, okay? So that's the negative feedback loop here. That's what we're talking about. So, say which one? Childbirth is one of the positive ones. Breastfeeding is another example. Blood clotting is another one. All right, there's a couple, but there's many more negative feedback loop systems in the body than there are positive feedback. Yeah, that's more, negative feedback is much more common. All right, so this part, when I go over the other hormones, this part is pretty much going to be the same across the board. Just change out the name of the hormone for whatever we're talking about, okay? So what I mean is the receptor is mainly going to be in the hypothalamus, okay? And it's going to respond to whatever any of those listed stimuli are there, okay? What time of day it is or whatever, all right? The control center is the same thing, all right? Well, I shouldn't say the same thing. The control center involves the hypothalamus and the pituitary gland, okay, for most of these hormones. In this case, all right, the hypothalamus is going to release growth hormone releasing hormone if growth hormone is too low, all right, or if it's early in the morning, that's when your growth hormone release time is usually pretty high, or if you're really young, okay? So here's what happens. All right, so growth hormone gets released, all right, from the anterior pituitary. It goes into circulation here, all right. Some of it, you can see here, it has two choices. Some of it, all right, will bypass the liver, and some of it will go into the liver through the circulation. If it goes through the liver through circulation, it'll stimulate the release of insulin-like growth hormone, okay? So you can see there's a couple different things that are going on here. We have our growth hormone. Point being is, all right, it goes into circulation here, okay? And it will affect its target cells. So what are the target cells? Yeah, I got an easier answer for you. All the cells. All the cells in the body are target cells, okay? But yeah, bone, skin, muscle, adipose tissue, everything, okay? All the cells in your body have receptors for these hormones, all right? So some of the effects, all right, are listed here, and I just kind of went through them for you, but I'll just kind of make it a little bit easier here, okay? So you can kind of see, and it's tough to see, all right? So you can see the effects on bone, muscle, and all the cells are growth hormone, increased growth, okay? It's going to increase, all right, cell division, all right? And to do that, all right, if you're going to build a house, you need energy and supplies for that. So what does that mean? It means that we need more amino acids for gluconeogenesis, okay, and for protein synthesis, right? Because amino acids are the building blocks for proteins, all right? We also need, all right, glucose. So what's going to happen in that case? We're going to have any process that can dump glucose into the blood, we're turning on, okay? So glycogenolysis, right, we just talked about that. gluconeogenesis, and lipolysis or lipolysis. All right? Okay? So that's what we're seeing here. Our liver is going to engage in glycogenolysis. It's going to release that stored glucose, and then it's going to make new glucose from non-carbohydrate sources, amino acids, proteins, fatty acids, that sort of thing. All right? While it's doing that, it's going to shut down or decrease glycogenesis, the creation of the stored form of glucose. Okay? Meanwhile, in our adipose tissue, we are going to increase lipolysis. We're going to break down fats, all right, and release them into the blood. Okay? And we're going to decrease the creation of fats, all right, triglycerides, all right, anything like that. Okay? Questions so far? That's a lot, right? It's not easy. All right, you guys were awesome. I'm going to not pause the recording. I'm going to stop right there.